Hello, students, friends, colleagues, and fellow explorers in science. I'm Enoch Yeung. I'm an associate professor in the Department of Mechanical Engineering. Thank you, Lena, for that warm introduction. And today I'm going to be talking to you about how genes communicate through twisting, the story of supercoiling in DNA. So to begin, I want to talk a little bit about what my lab does and introduce you guys to what our work is focused on. We study at the in a single sentence, we study genetics in bacteria. And so what you can see here pictured is an electron micrograph of E. coli bacteria. And you can see the scale bar on the bottom right corner there that shows that it's roughly 2 microns in length, or 1 to 2 microns in length. And E. coli is an example of a bacteria that lives where? Does anybody know? In our intestines, that's right, that's right. So anyhow, so E. coli bacteria is a very common bacteria that we study in university settings and has very important applications in probiotics and biotherapeutics and generally in our understanding in science. Bacteria are extremely small. Let's see if I can get this to advance. This is a diagram of what a bacteria might look like. It's about 1 to 2 microns in length. And does anybody know what this is? Good guess. A tick is about the same length or scale, and that's what we're trying to get at with this slide. This is a carpet beetle. It's a little tiny bug that sometimes you see crawling across your carpet. They're white and black. They're pretty harmless, as I understand, unless you have a huge infestation of them. And they're usually 2 to 3 millimeters. So this is about 1,000 times the length of a little bacterium. All right? And so just to give you a scale of the things that we're studying, this is a slide to do that. And why do we want to study bacteria? Bacteria are found everywhere. They live on our skin, they live on our lungs, they live on our gut, on our food, and our pets, to name a few examples. There are many, many more. Now, as scientists and engineers, we seek to understand the dynamics of and harness and control the dynamics of bacteria for the betterment of society. And part of that is understanding how living processes work naturally, but also part of it is understanding how we interact with bacteria in the world around us. Now, raise your hand if you've ever interacted with bacteria before. Everybody's hand should be up. And why is that? Well, because if you have ever done any of the following things, you have changed the dynamics of bacteria somewhere in the world, probably in yourself. All right? So if you have ever drank probiotics, you have changed the growth dynamics of bacteria in your gut. And that is because there are bacteria all over your gut lining. And every time you take one of these probiotics, or any time you eat in general, you are nourishing or potentially starving or attacking or supporting some group of bacteria in your gut. And some modern science has shown that our health as human beings, our well-being, our mental health, our capacity to remember things is strongly correlated to our gut health and the microbiome, the collection of bacterial species that constitute our gut, okay? Now, if you ever put on lotion or sunscreen, you have changed the dynamics of bacteria on your skin. Staphylococcus epidermis is an example of a bacterium that lives on your skin on a regular basis and is naturally there. It's present. It's part of a healthy skin dynamic. If you have ever brushed your teeth, hopefully you've all brushed your teeth, but in case you have forgotten and you have also forgotten to brush your teeth, you have changed the overnight dynamics of your oral bacteria, right? What happens every time we brush our teeth? We basically onslaught the bacteria in our mouth. We reset the clock. We say start over again, and oftentimes wipe out large populations of bacteria. Now, certain species of bacteria have evolved in the gum line, that have evolved in the bloodstream local to our gums to be present, to persist despite these regular diurnal activities. But nonetheless, please note there are massive fluctuations in the number and the density of bacteria in our mouth every time we brush our teeth. So think about that tonight. All right, if you have eaten fresh vegetables, especially if you have eaten it from the garden without washing it. You have changed the collective composition of your oral bacteria. There are plant-promoting and plant adverse bacteria all over plants. And plants utilize bacterial function to be able to accumulate specific metabolites, to be able to balance themselves relative to the ecosystem that they live in. And so if you have a garden and you're eating vegetables from your garden, if you're sampling carrots or peas, or even if you're taking a fruit from a fruit tree, generally speaking, you're going to be sampling some bacteria and even potentially fungal species that are potentially helpful, neutral, or harmful to your gut. One more example. Raise your hand if you have a pet. Okay, about half of the audience, it looks like. It's kind of hard to see through the lights. So it looks like about half the audience. But if you have, raise your hand if you've kissed your pet before. You don't have to disclose if you don't want to. All right? So if you have kissed your pet, you have changed the collective composition of your oral bacteria. All right? And so why is that? Because on the hairs, on the skin, on the saliva of your, in the saliva of your pet, there will be a different collection of bacteria that are living there. So hopefully I've shown you with these examples how bacterial populations are really persistent. They're ubiquitous all throughout our human experience and through life. Bacteria we often label as the villains. We say, oh, they cause disease. You may have heard of things like MRSA. You may have heard of things like Pseudomonas, organisms that are bacteria that specifically have pathogenic properties, that they can create disease in the human being. But there are so many other species of bacteria that are persistently present in the environment, that are simply neutral in the environment, or that are helpful to human beings and for our overall health. So understanding them and studying them is ultimately the key. Marie Curie was famous for saying, and you may have heard the quote before, nothing is to be feared, only understood. And so that is our aim. We want to study bacterial genetics so we can understand them. Now in the lab, this is an example of a simple experiment that we can do. So Lena, if you can cue the movie, I can see if I can get it to go. Perfect, thank you, Lena. So in the lab, we place bacteria under a microscope and label them with fluorescent markers that allow us to study the activity of specific genes. So what you see here is a picture from top down, looking as if you're looking from the top down on the bottom of a plate. This plate has a sandwiched set of glass. So there's essentially, you can think of it as two sandwich layers of transparent material, but you can think of them as glass essentially. And inside, there's a gap, and that gap is regulated by these little columns right here. These little black marks here are all individual columns that guarantee an evenness in height of this little chamber. What we do is we then grow bacteria from our freezer. After we have recovered them for a period of time, we then put them into a port on the right side of the plate. And on the left side of the plate, we engage a vacuum so as to create negative pressure, drawing the cells through and into the chamber. The chamber's height is precisely calibrated to be roughly the height of half of a bacterium. So that way when the cells come through, they get trapped. And as they get trapped, we can then observe how they're going to grow as we flow nutrients, media, metabolites for them to consume. All right? We can do this with any kind of bacteria that is willing to get into this trap. All right? And that's a lot of them. Okay? But furthermore, what we can do is we can take the bacteria and we can modify their genetics using tools from synthetic biology, molecular biology, microbiology. We can modify the genetics of these bacteria and label specific genes with fluorescent proteins. How many of you have heard of green fluorescent protein? Great, so about 50% of you, all right? Maybe the more biology-oriented students in the group. And if you haven't, that's fine. But green fluorescent protein is a protein that was harvested from a species of bacteria in the ocean from the bobtail jellyfish, cuttlefish. And what they found was that this bacteria naturally produces a fluorescent protein that is, upon exciting at a certain wavelength, results in fluorescence. The green fluorescent protein has since been isolated and now is used as a common tool to visualize fluorescent gene expression in any sort of bacterium. So what we have done here is we have labeled two genes. One gene is associated with biofilm formation, and the other is an ancillary gene that is opposed to biofilm formation in bacteria. I'll talk about biofilms in a second. But the point is, we study the genetics of bacteria. We're very interested in understanding, as a lab, how do we track how genes are expressed across a population of cells. So think of that as one dimension, from one individual bacterium to another. And separately, as a function of time. As a function of time, all right? A third axis is across the genome, across all of the different genes in a bacterium. Okay. So, the more scientific way to pose this question, and the question you'll see if you search my website, which I'm sure some of you have, is this question right here. This is the guiding question to the majority of the research that we do in our lab. I will also note that we do research in machine learning for the purpose of understanding biology. We do research in control systems engineering because we're ultimately trying to control certain aspects of biological dynamics. But the core question, I would say, that drives about 50-60% of our research, the majority of our research, is how do mechanical forces in DNA shape gene dynamics and cell fate? How do mechanical forces in DNA shape gene dynamics and cell fate? Raise your hand if you know what the word DNA stands for, or the abbreviation DNA stands for. So it looks like about 3/4 of you, maybe like 4/5 of you. So we won't spend much time on that in terms of what that means. I'm assuming you have some exposure to it. But what you see here, this picture, is 3 separate time series movies, but I've broken them up so that way you can see what happens at 5 minutes, 20 minutes, 40 minutes, 60 minutes, and so forth. All right? And what you can see is the division of cells across 3 rows. And what is really probably not so easy to believe is that all these cells contain the same 2 genes. A gene that produces a green fluorescent protein, hence the color green that you see, and a separate gene that produces a red fluorescent protein. The same 2 genes. So what's different about row 1, 2, and 3? The primary difference between these individual rows is the mechanical forces in the DNA surrounding these genes. Otherwise, the same 2 genes that are in this row are present in this row as well, and are present in this row as well. All we did was change the relative gene orientation, and directly as a consequence, the mechanical forces in the DNA around the genes. All right? And what happens as a consequence? The populations of the cells have a totally different phenotype. Phenotype is a scientific word used to describe the physical manifestation of a biological organism. All right? A physical manifestation of what? The genetics of an organism. And so we can see here, by simply changing the mechanical forces around genes, not the actual gene sequences themselves. We're not mutating the genes to make one brighter or stronger. We're keeping those things constant. We're changing mechanical forces. We can see a direct correlation with changes in how the cells ultimately respond over time. All right. So this is my lab, a little bit of in terms of lab personnel and roster. I am very, very grateful for a talented team of postdocs, PhD students, master students, undergraduates as well, and occasionally high school students that have helped to bring about some of the things you're going to see in terms of research. Today's talk is very much geared towards a general audience, so we're not going to get super technical, but if you want to continue to have technical conversations, quantitative conversations that involve lots of scary math, please feel free to reach out to me afterwards. But I will comment here, just as a shameless solicitation, that we do run a annual summer workshop that's funded by the National Science Foundation. Here is last year's cohort, and they did incredible things with CRISPR engineering and DNA supercoiling. Now, I should say, if you are interested and want to participate, feel free to reach out to me at this email. You can do so. Continuing on, like I said, our lab studies genetic code in bacteria. And this is a picture right here, a transmission electron micrograph of E. coli. And what you can see here is compacted nucleoid DNA. The chromosome of bacteria generally is found in a compacted section of the cell and accumulates like this. Now, how long is the average chromosome of a bacterium? Does anybody know? It's anywhere from 1 million to 10 million base pairs, usually, in range. For this species of bacteria, it's about 4 million base pairs of DNA. Now, how in the world do we fit all of that into 1 micron, 1,000th the length of a carpet beetle? And the answer is with DNA twisting. All right. So, just as a quick reminder, DNA is a double helix. All right. And you can see that here. 2 strands of DNA interwoven, right, with complementary base pairs. Adenine, thymine, guanine, cytosine. Adenine and thymine bind together to form AT bindings. Guanine and cytosine bind together to form CG bindings, right? And this forms the template or genetic code of life. And I'm here to tell you today that mechanical forces on the DNA molecule itself have a very big role to play in terms of how RNA is transcribed from DNA and ultimately then how protein machinery is produced from the RNA. The very first step starts here, and that is where a lot of the action that we're going to be focusing on is happening. Now, again, the structure of DNA is a double helix. So what does that mean? It's naturally twisted. It is naturally twisted, okay? So because DNA is twisted, what this means is that any strand of DNA is going to have a certain number of times that it twists around its axis. So if you imagine the DNA as a long linear strand, all right, how many times are those 2 strands going to turn around on each other, all right? That is called the linking number of the DNA, all right, if we want to get technical. And the degree of twist in DNA is conserved with the degree of writhe. As you increase the number of times that the DNA molecule is wrapping around itself, all right, what happens is that there's a point when the bonds can no longer support twisting on a planar fashion, and it starts to twist on itself in 3D space. That form of twisting we call writhing of DNA in the 3-dimensional space, all right? Here you can see an electron micrograph of a relaxed state DNA. This is a plasmid, a circular fragment of DNA, the common currency that we use to manipulate genetics in bacteria. And here on the right-hand side, you can see supercoiled DNA, DNA that has been exposed to excessive amounts of twists or has accumulated excessive amounts of twists and now is starting to writhe on itself and twist on itself. And that is precisely how DNA can be compacted into a cell. Continuing on, twist can be a good thing. I should note that garden hoses, if you have one, are a great way to explore the principles of twist and writhe. If you stand with your brother or sister and you twist the garden hose, you can see after a while the garden hose will form these lovely knots that make it so that your parents or your gardener loses their temper when they're trying to water their vegetables. But nonetheless, DNA is twisted, and here are some more pictures of DNA twisting. So this is mitochondrial DNA, supercoiled from plants. Look at that spaghetti-like structure. Isn't that interesting? Over here on the right-hand side, we have supercoiled DNA. It's about a 4,000 base pair plasmid, and it's expressed in saline buffer. This was published in a Proceedings of National Academies of Sciences article in 1997 in which they were looking and seeing how the structure of DNA responded to ionic changes in the environment. Okay, now what we are studying again is bacterial dynamics, and we're interested in mechanical forces in DNA specifically because what we have recently shown, this is an example of an older article that has studied this question, is that twisting in DNA facilitates communication between genes. So, we've established that DNA is double-stranded, that it's helical in nature. It's twisted, all right? What happens if I have two genes that are encoded, one going this way in its directionality, and the other on the other strand going the other way? When you try to produce an mRNA molecule, what happens to the DNA? They have to open up. They have to separate. As they separate, what happens? The twist that was conserved and present here in this section now has to go elsewhere. So what we get is the formation of writhing or hypertwist or supercoiling downstream of the gene of interest in this case and downstream of the gene of interest in this case. Now if you take a rubber band and you twist it so it looks like a double helix, then you have a friend stick their finger in between and pull apart a section in the middle, what you'll see is that there are two types of twists that form. Negative twist that follows your left hand, so holding your left hand, all right, it's going to twist this way, and positive twist that's going to form the other way following your right hand. We call it positive and negative supercoiling. They are opposing each other in direction. They twist in opposite directions. So every time you open up DNA, you get negative upstream and positive downstream. And that's true even if the transcription is occurring this direction. And so what happens? Between two genes, you're going to have, what we found in this paper was you can have twist that is going to cause positive coiling one direction, where the DNA loops this way over and then under, and then now we reached a section where there's negative coils, where it's under and then over, under and then it's over, so we have opposing forms of twist. It's a mechanical basis in which genes can talk to each other. A mechanical basis, all right? If you want to get the attention of your younger brother, or I should say vice versa, usually it's your younger brother or younger sister who's doing this to you maybe. If they want to get your attention, one of the ways they can do it is they can twist your finger, right? How effective is that at getting your attention, at communicating to you? It's almost instantaneous, right? Ow, please stop twisting my finger. Don't do that, right? Twisting is a very, very effective way to transmit information from one place to another. I'm not recommending that as a form of communication among human beings. But I am saying that what we have found from a scientific standpoint, from a genetic standpoint, is that twisting actually causes communication between genes. Now let's look at this a little bit more. Let's consider now all three possible scenarios that we might encounter when we look at just two genes. As we consider 3 genes, 4 genes, 5 genes, the number of possible combinations of ways that things can talk to each other increases combinatorially, all right? But if we look here, we can see that convergent genes are going to produce a domain of positive and negative twist, where transcription is driving that twist against each other so that there's this sort of opposing logic, all right? In the divergent case, where genes are transcribing away from each other, what happens is this twist is generally less intensely accumulated, but there will generally be one side that's going to be favored more than the other. And then in tandem orientation, what we have is this weird cancellation effect, where one gene really affects the other, but the other one is not so much going to affect this one, all right? So different arrangements have different consequences in terms of bacterial dynamics. So coming back here, we can see convergent orientation does what? It produces my way or the highway kind of dynamics. Red or green, but not both at the same time. All right? Divergent produces asymmetric dynamics favoring the red gene. And tandem orientation produces correlated expression of both genes, but ultimately a cassette that favors green expression. Changing the twist states around 2 genes changes the makeup of the entire population. So in particular, one thing that we've been studying and that is currently unpublished in terms of our research. We've just discovered this, but I wanted to show you guys some of the cutting edge stuff. Please don't scoop us, obviously. One of the things we've discovered is that in biofilm clusters in naturally occurring pathogenic bacteria, you can take the biofilm clusters and study their DNA twist state. And what we have found is that biofilm cluster formation heavily leverages a convergent orientation. What does that do? Well, it makes it so that some of the genes are going to grow rapidly, or some of the cells are going to grow rapidly, while others decide to form biofilms. They will divide and conquer. All right? Pretty effective. All right? So that's pretty cool. And this example here is a picture of Staphylococcus aureus in a catheter. All right? Now, we want to understand the statistics and the mechanics of why this happens. So, Lena, if you could start this movie. This is a picture of molecules that are tethered on glass that have DNA twist and writhe, all right? The writhe is depicted by these little flashing dots here. You can see that's accumulation of DNA, all right? The length of these DNA are 20,000 base pairs. If you've done PCR, this is a very difficult PCR reaction to get working. I promise you, you do not want to try this. All right, it's a very difficult PCR because amplifying that long of a fragment of DNA is fraught with difficulty. But nonetheless, we managed to get to work, all right? And what we can see, thank you, Lena, is right here is the setup for how you set up something like this. You can take an antibody that binds to its cognate, and what you can do is attach that to a DNA strand. The DNA strand normally, historically, has been something that doesn't have any specific genetic code, but in our case, because we love bacterial dynamics, we love gene expression, we're interested in control, we have actually included promoter sequences, protein sequences of fluorescent proteins. Now what you do then is on the other side of this, you attach a small magnetic bead. A small magnetic bead that's about 0.3 microns in diameter. So really, really small, okay? And what you do is then you can put a magnetic field around this whole setup, all right? So here's an example or a diagram that might show what we're doing, all right? And you can see here you can form a magnetic field, and as you do so, you can introduce artificially a certain degree of twist in individual DNA molecules. And I kid you not, we have had high school students playing with magnets in our lab, controlling individual DNA molecules tethered to glass. It's a really cool feeling to know that you have that degree of control at your fingertips, all right? Now, we are at the point now where we get constellations of DNA molecules. The green here indicates fluorescent polymerases. The red here indicates those plectinemes. And what you can see is ultimately that we're at the stage where we can study transcription, DNA structure, DNA repair, all at the same time. Now I'm out of time, so I'm going to switch here to the conclusion talk, or the conclusion slide. And I want you to think about what is worth remembering from this talk. Number one, bacteria are really an important part of our world. Some help us stay healthy, while others help plants to grow, and yet others cause disease. Secondly, bacteria are programmed by genetic code and DNA. Thirdly, DNA is a double helix with twist and writhe properties. And most importantly, hypertwisting or undertwisting is a way that genes can mechanically communicate with another on the same DNA strand. The twist state changes the way in which important genes are expressed, which then changes the fate of bacterial cells. Under high magnification microscopes and single molecule studies, we have begun to unravel the dynamics of DNA twist and gene expression. Now, if you're interested in learning more, feel free to reach out to me via email. Thank you for your time and attention. I'd be happy to take any questions.